You're welcome to my another session of cytology class. In this session, we are going to discuss on the topic cytological proof of crossing over. Morgan and his collaborators established the genetic basis of crossing over and linkage described in the exchange of parts between the homologous chromosomes and linear arrangement of linked genes along the chromosomes. This genetic inclination or bias could not be demonstrated cytologically since we cannot observe the homologous chromosomes being all identical under the microscope, and because of the reasons like crossing over occurs between the homologous chromosomes, and such chromosomes are alike in appearance, and it is not possible to distinguish between them in a microscope. And secondly, during crossing over, the four chromatids are intimately coiled around one another. And third aspect is, in living cells, crossing over cannot be seen. In fixed and stained cells, one cannot say that chromatids have exchanged parts or not. For nearly 20 years, crossing over remained only a working hypothesis with the genetist. Finally, the cytological evidence which was established beyond doubt the occurrence of crossing over and was given by Stone on Drosophila and Creighton and McClintock on Maize. What do you mean by crossing over? Crossing over may be defined as an interchange of chromosomal parts between non-sister chromatids of homologous pair of chromosomes resulting in the recombination of genes at meiosis, prophase 1 and in diplotin stage. And it takes place during the diplotin after the homologous chromosomes have undergone four strand stage, that is the four chromatid stage. The word crossing over was coined by Morgan in 1912. Morgan and his collaborators established the genetic basis of crossing over and linkage describing the exchange of parts between the homologous chromosomes and linear arrangement of linked genes along chromosomes. But the cytological evidence was established about the occurrence of crossing over was given by a system on Drosophila and H.B. Creighton and McClintock on Maize. Now, Stun's experiment on Drosophila. Curt Jacob Stun in 1931 by direct cytological evidence demonstrated that crossing over involves interchange of parts of homologous chromosomes. The female Drosophila carries XX chromosome and the male Drosophila carries one X chromosome and one Y chromosome. He made two X chromosomes of the female that are different from each other. X chromosomes by treating such flies with X-rays. One X chromosome had a part of a Y chromosome attached to one end, whereas the other X chromosome has a broken into two unequal segments. Thus, both aberrant X chromosomes were cytologically detectable. The broken X chromosome contains a recessive gene CAR for carnation eye color and a dominant G capital B for bar eye shape. There is one fragment having both the genes. While its homologue X chromosome contains the dominant gene CAR plus for red eye color, the recessive gene B plus for round eye shape. The female flies Heterozygous for these two morphologically distinguishable X chromosomes were produced by crossing. And these heterozygous females with a trans configuration were crossed to the males with carnation eye color and round eye shape. The genes they considered are CAR, that is recessive for carnation eye color, CAR plus dominant for red color, capital B dominant for eye shape, and B plus recessive for round eye shape. This is the diagrammatic representation of the Stun's experiment on Drosophila. The female fly with two X chromosomes, one X here it is broken, that is the abnormality, and in this X chromosome another abnormality at one locus of this chromosome, and this is carnation with bar eye shape, and this one is red color with round eye shape. No crossing over, and here it is crossing over which occurs during meiosis. So at the end, four gametes are produced. This is the gamete produced by the male fly, that is the sperm. This is another sperm. Their fertilization by the sperm from carnation F1 male. Same thing here also. But at the end, four individuals or four kinds of offsprings are produced. One with carnation bar, red round, carnation round, red and bar. So these two are the parental combinations of both the genetic traits and chromosome abnormalities, whereas these two, they are the recombinant combinations of both genetic traits and chromosome abnormalities. So the crossing over was indicated phenotypically, showed microscopic evidence of exchange between the homologous chromosomes. So this proves that the cytological basis of crossing over, and it was established in this experiment. Another experiment that is was conducted on maize, and this was conducted by Harriet Creighton and Barbara McClintock. Harriet Creighton and Barbara McClintock in 1931 demonstrated the correlation between the genetic crossing over and the exchange of parts of homologous chromosomes. And they analyzed crosses involving two loci on chromosome number 9 of maize. And the genes represented are capital C colored seed, small c colorless seed, capital W small x starchy endosperm, whereas small w small x represented waxy endosperm. And they made use of a chromosome with a densely stained knob at one end and an extra translocated piece of chromosome at the other. They created a heterozygote with the following characters, that is the repulsion, configuration of genetic markers and cytological landmarks on both the ends of one chromosome. And they next perform a test cross to this stock with small c, w, small x, small x tester. The plant heterozygous for colored aileron and starchy, that is non-waxy endosperm characters, and carried these genes in repulsion phase, that is capital C, small x, and small c, capital W, small x. So capital C, small w, small x was carried on the knobbed chromosome and small c, capital W, small x on the knobless chromosome. Such plant was test crossed with the plant heterozygous recessive for both the characters, that is colorless and waxy. This is the diagrammatic representation of Creighton and McClintock experiment on maize to prove the cytological evidence of crossing over. So they performed a test cross with a cytogenetic stock which is heterozygous, and this individual has colored seeds with starchy endosperm, whereas this one colorless with waxy endosperm. This is the tester, and this one is the cytogenetic heterozygous stock. So the crossing over involves the physical exchange of the chromosomal material and then the recombination of phenotypes. So they obtained a parental phenotype. This one is the recombinant phenotype. Again a recombinant phenotype. This is parental phenotype. So from this work, it could be conclusively stated that the crossing over involves the exchange of genetic material between the chromosomes.